So, good morning, good afternoon, everyone, and welcome to this session dedicated to a topic that is both highly relevant and increasingly complex: toxicological risk assessment and the analytical strategy for control of nitrosamines. Before to start, just a few technical details. This webinar will be recorded and the presentation will be shared with all the participants afterwards. Furthermore, a Q&A session is scheduled at the end of this presentation. And for any question we are unable to address during the session, written answer will be provided in the following days. My name is Alessandro Lombardi, and I'm the head of Pharma Division at Neotron, that is part of Cotecna Group. Let me spend just a few words about Cotecna. Cotecna is a global provider of testing, inspection, and certification services across 5 key sectors that include food and pharma, agriculture, metal and minerals, certification ESG solution, and consumer goods. Neotron is one of Cotecna's flagship laboratories based in Italy. It brings over 15 years of experience in testing field. We are specialized in the food supplement and baby food testing, food contact material, cosmetics, and of course, high-end analytical support for the pharmaceutical industry. In the pharmaceutical sector, we support our clients throughout the entire product life cycle, from raw material to finished drug product, with a broad portfolio of GMP certificate services. Of course, we are authorized by the Italian Medicine Agency, and we were also successfully inspected by FDA twice so far. Our services include extractable leachable studies, together, of course, with the nitrosamine, that is the topic of today's webinar, elemental impurities, organic impurities, and as well as method development and validation. So let's go to the agenda of today. As many of you know, since the discovery of nitrosamine in sartan in 2018, what started as a single product alert has escalated into a global industry-wide concern. Regulators now investigate this compound closely due to their carcinogenic potential, structural diversity, and most importantly, their unexpected presence in a wide range of finished drug products. To help us to navigate this complexity, we are joined today by 2 experts who will guide us through both the analytical and toxicological dimension of this challenge. First, Caterina Cappuccini, that is the quality control manager at Neotron Pharma, will present a talk titled Nitrosamine Impurities from EMA Update of Nitroso API to Real Case. She will share how analytical laboratories are evolving their testing strategy in response to the latest guidelines, supported also by real case that she will present. Afterwards, Manuela Pavan, principal consultant at Innovatune, will delve into toxicological risk assessment of nitrosamines, illustrating how toxicological evaluation, when integrated early in the process, can support both regulatory submission and overall risk mitigation. Now, let's go with the first presentation of Caterina, that will be followed by Manuela's presentation. Thank you.
Thank you, Alessandro, for your introduction. Let's jump now in the topic of today, nitrosamine. And let's start a little bit from the beginning. What is a nitrosamine? A nitrosamine can come from the reaction of a secondary amine present in the API and a nitrosyl source that could be present in excipient. The nitrosamines can be potential carcinogenic and mutagenic potential, and this is the big awareness from all the regulatory. Next. The interest in nitrosamine started after the alarm of NDMA detected in valsartan in 2018. At the beginning, all the focus was on small molecules, mostly arising from solvents and reagents, potentially potent carcinogenic. In 2020, EMA releases a nitrosamine impurities guideline. Now in April 2025, was released the last update. If at the beginning the attention was on small molecules, now is more shifted on nitrose API related, large size molecules, drug substance like structure, mutagenic, potential unknown. Next. Several are the guidelines published from different authorities. We have the EMA, Government of Canada, or FDA. Next. The document question and answer of EMA give indication on several requirements. For example, question and answer number 9 explain the requirements of the analytical methods. An analytical method has to demonstrate to be sensitive, accurate, and repeatable. We can find also the definition of the limit of quantitation that provides the minimum level at which an analyte can be quantified with acceptable accuracy and precision, and should thus be used for impurity testing and decision making. In the case, for example, of routine methods, the limit of quantitation has to be equal or below the acceptable intake. In the case of skip testing, it has to be between the 10 to 30%. In the case of omission of specification, the limit of quantitation has to be below the 10%. Next. Another important document is the Appendix 1 that contains all the nitrose API, large molecules, and the nitrosamine, small molecules that has to be controlled. Now more than 200. The acceptable intake of each nitrosamine is a fundamental information to calculate the limit of specification. This limit of specification is calculated dividing the acceptable intake of each nitrosamine for the maximum daily dose of the API. The 10% of the limit of quantitation we were talking before is the 10% of this limit of specification calculated as just explained. Next. But in what way we can support? So toxicological and risk assessment aspect will be explained by my colleague after my presentation. From the laboratory side, we can support in method development of API and drug product specific, stability studies, so drug product quality and shelf life, evaluation of interaction of API and excipients. What is the general approach? So we receive the customer request, we check the availability of the standard, we develop the method, we validate the method. Next. The availability of the standard is a very crucial aspect and plays a very big role. We collaborate with a specialized company that have in stock or they have to synthesize the standard for us. Sometimes can take also 2 months to get the standard. So when we have the standard, then we can start with the development of the method. Next. It's very important that when we receive the standard, the supplier gives all the information about the standard. So the characteristics, the solubility, the mass spectra to compare to the one that we obtained during the setup in our laboratory, and the indication about chromatography are very important because it's a good starting point for the setup of the method. Next. When the standard cannot be synthesized, maybe other paths are available. For example, in this case, the customer asks to investigate the possibility of having nitroso-disulfiram in their product. The supplier could not isolate the nitroso derivative API because the API is not stable under acidic condition and transform directly in their correspondent tiols. In this case, nitroso-diethylamine. From the collaboration between the customer and the supplier, what we have validated in the end was the nitroso-diethylamine to check the possible formation or the contamination in the sample. Next. Once we have the standard, a suitable analytical technique has to be chosen, usually LCMS because it's the most sensitive one. We optimize all the mass parameters and chromatography, so molecular ion characterization, chromatographic column, mobile phases. The gradient is very important in order to separate the API and the nitrose API and all the mass parameters in general. A very critical step is also the sample preparation because depends on the pharmaceutical form. And it's very important to find the current solvent of dilution in order to not having false positive. Next. At this point, once the method has been developed, we have two paths. Quantitative method, that is usually the common way, or a qualitative method that can give a clue, a general info, let's say, on the entity of the contamination. Depending on the results obtained, we can choose if proceed after qualitative method, if proceed with a validation or not. Usually, if the sample is free of contamination or the contamination is below the 10% of the limit of specification, the documentation collected is already enough for the regulatory, and because we are in the case of omission of specification, so below this 10%. Next. Now I will explain three different case studies that are connected to each other. Next. So the first case study is high content of nitroso-nortriptyline was highlighted in the finished drug product above the limit of specification. With the content very high, is difficult to validate the method and challenge the method at the limit of quantitation level. So we have validated the method on the placebo without the API. The method was validated with expanded linearity and wide accuracy range. So in the case of positivity, we have a method that cover high results. The method validated then was verified on the finished drug product and the API. For verification method, I intend like include the accuracy at verified accuracy at limit of quantitation, at the limit of specification, and the repeatability of the method. Next. To understand this high contamination found for this finished drug product was this finished drug product was then studied the interaction of the API and some excipients. We have studied the same API but from different supplier. The nitrose API was not detected in the API itself, and these two different API were studied with two different excipients, stress and no stress. What we want to highlight with this experiment is that different excipients can contribute differently with the formation of nitrose API, but not only the excipients, because we have noticed that with the same excipients, different results have been obtained with different API supplier. So this is can be also an important information that can help the customer in the choice of the supplier. Next. On the finished drug product was then performed a stability study to monitor the content of the nitrose API, and this is a very good approach in order to evaluate also the quality and the shelf life of the finished drug product. So to conclude, next. To conclude my part, so the regulatory evolution encourage process improvement. Process improvement needs an analytical support, and the analytical support is the tool to ensuring safety of pharmaceutical drug product. Now I leave the word to Manuela. Thank you.
Thank you, Caterina, and thank you all for joining the webinar. So now we will focus on toxicological risk assessment of nitrosamines, and I will focus on those challenging cases where substance-specific data are not available. So we will try to cover the scientific methods and the regulatory strategies used in this assessment. I will recall some concepts that were just now introduced by Caterina. So nitrosamines are a broad and structurally diverse class of compounds that are characterized by the presence of a nitroso group attached to a nitrogen atom of an amine. They typically form through the nitrization of secondary or tertiary amines and are widely studied because of their genotoxicity and their recognition as potentially high potent carcinogens. Next. Nitrosamines are generally classified in the two primary types. First, we have the small, the alkyl nitrosamines, such as NDMA or NDEA, and these traditionally have been extensively studied in rodent carcinogenicity studies, so they have a lot of data. While on the other side, we have the nitrosamine drug substance-related impurities, the NDSRI, and these are more complex drug-specific compounds with typically limited toxicological data available, making them data poor, and as a consequence of that, more challenging and difficult to assess from a toxicological perspective. Next. The ICHM7 guideline provides a framework for assessing DNA reactive mutagenic impurities, and a key concept in the guideline is the cohort of concern, which refers to highly potent mutagens for which the concept of the threshold of toxicological concern does not apply. So in these cases, the default limit of 1.5 microgram per day is considered insufficient, and the case-specific toxicological risk assessment is needed. The cohort of concern includes structure classes such as the aflatoxin-like compounds, the nitroso compounds, the alkyl toxic compounds, so substances that are so potent that even very low exposures may pose a significant carcinogenic risk. And the nitrosamines belong to, therefore, a broader group known as the nitroso compounds, which all share a common structure feature, so the nitrogen atom bonded to another nitrogen that is double bonded to an oxygen. While both nitrosamines and other nitroso compounds fall under the court of concern, the current regulatory efforts are primarily focused on nitrosamines. Next. As it was anticipated by Caterina, formation of nitrosamines is possible in the presence of secondary, tertiary, or quaternary amines and nitrite salts under acid reaction conditions. In these specific conditions, nitrate can generate nitrous acids which can react with the amine to produce a nitrosamine. This type of reaction, which is known as nitrization, is a key concern in pharmaceutical manufacturing. And any process that involves both nitrates and amines in an acid environment carries a risk of forming nitrosamine impurities. Next. Key sources of secondary, tertiary, and quaternary amines that can lead to the formation of a nitrosamine include the API itself, its degradants or raw materials that contain secondary or tertiary amine groups, tertiary and quaternary amines that are intentionally added during the synthesis, so for example, reagents or catalysts, and certain amide solvents which can degrade into secondary amines under specific conditions. On the other hand, nitrates, the main nitrosating agents, may come from a variety of sources. They've been detected in many excipients, often as part per million levels. Other sources also include potable water and container closure systems, such as the packaging materials and the manufacturing equipment. And given these multiple potential sources, a comprehensive evaluation of the manufacturing process is essential in order to identify whether a nitrosamine precursor may originate. Next. To understand the approaches underlying the toxicological risk assessment of nitrosamine, it's essential to first understand the mechanism that is responsible for their potential carcinogenicity. Nitrosamines require metabolic activation to exert their genotoxic effect, and this slide illustrates the key steps in the metabolic pathway that can ultimately lead to carcinogenic outcomes. So the first and the critical event is the cytochrome P450 mediated alpha hydroxylation, which generates an unstable intermediate. This intermediate rapidly rearranges into a highly reactive diazonium anion that is able to alkylate the DNA. This DNA alkylation may be repaired by the cell's DNA repair system. However, if it is unrepaired, it can be propagated during DNA replication, leading to mutations. And over time, the accumulation of such mutations may contribute to the cancer development. Next. So the nitrosamine bioactivation is an indirect process. This is very important. That requires metabolic conversion, metabolic activation by liver enzymes and potentially enzymes for other tissues to generate the ultimate carcinogenic activity. And the critical step, therefore, is this P450 catalyzed alpha hydroxylation, which initiates a series of reactions that are leading to the formation of a diazonium anion that is then alkylating the DNA. This mechanistic understanding explains why nitrosamines are potent carcinogens across a wide range of animal species, including humans, and most organisms have the necessary enzymes to activate them. It also guides regulatory approaches such as the structural activity relationship-based risk assessments, and compounds, for example, that are less prone to undergo the alpha hydroxylation are generally considered to be or to pose a lower concern. Next. Understanding the mechanism of nitrosamine bioactivation is essential, not only to explain why these compounds are widely carcinogenic, but also to provide a solid scientific foundation for assessing how structural variations influence their potency. So building on these mechanistic insights, the literature has identified several structure features that can tune, that can modulate the carcinogenic potential of N-nitrosamines. And these structure activity relationships, or SAR features, include pharmacokinetic factors. For example, carboxylic acid groups may promote faster clearance from the body, and this can reduce systemic exposure. Steric hindrances, so bulky groups near the alpha carbon, can physically block the cytochrome P450 enzymes, and therefore they can limit the bioactivation step. And also electronic effects, so electron withdrawing substituents can lower the likelihood of a diazonium anion formation, and this again can decrease the potential for the DNA alkylation. So these features help to explain why some nitrosamines are more potent than others, and are also key considerations that are taken into account in the scientific and in the regulatory evaluation of the risk. Next. As we mentioned before, this scientific foundation of the mechanism of nitrosamine bioactivation guides the regulatory approach, which is now broadly harmonized across major global health authorities, including the EMA, the US Food and Drug Administration, and Health Canada, for example. While there may be minor differences in the specific implementation or guidance documents, the core principles are basically aligned among different regulatory authorities. One key example is the carcinogenic potency categorization approach, the CPCA approach, and I will get back on this in a few minutes. Next. Before focusing on the regulatory context, just to put it into context, it's helpful to look at how nitrosamine impurities fit within the broader regulatory framework for impurities in medicinal products. So the non-mutagenic impurities are typically assessed under the ICH Q3A and Q3B, where the limits are in these cases based on general toxicity data, commonly from repeated dose studies. In contrast, mutagenic impurities, those that can directly damage DNA, are regulated under the ICHM7 guideline, which introduces the concept of the threshold of toxicological concern, which is a risk-based exposure limit below which there is a very low probability of the adverse health effects, even without a specific toxicity data. For most mutagenic impurity, the threshold is set to 1.5 microgram per day, representing a lifetime cancer risk of 1 in 100,000. However, there are some impurities, including nitrosamines, that fall into what we mentioned before is known as the cohort of concern. So a small group of compounds with particularly high potency, high carcinogenic potency. And for these substances, substance-specific limits are required when the data are available. In cases where the data are not available, a conservative class-specific limit is applied, and for nitrosamine, this was initially set to 18 nanogram per day by EMA and 26.5 nanogram per day by FDA. But more recently, these limits have been refined using a structure-based approach, the CPCA, the carcinogenic potency categorization approach. Before moving on that, let's first consider the case where the nitrosamine is data-rich, so when experimental toxicological data are available. When sufficient substance-specific animal carcinogenicity data are available for nitrosamine, the ICHM7 guideline recommends calculating the TD50, so the chronic daily dose that causes tumor in 50% of test chemicals, and using this to derive a compound-specific acceptable intake limit for lifetime exposure. For some well-studied nitrosamines, such as NDMA, NDA, extensive rodent carcinogenistic data are available, and as we said, this allows acceptable intake to be derived directly from the TD50 values. And the reliable TD50 values can be found in public domain, in publicly accessible databases like the carcinogenic potency database, the CPDB database. Next. When a nitrosamine is identified as being characterized by data poor availability. So when it's identified, but there are not enough substance-specific data to derive a compound-specific acceptable intake, the assessment becomes more challenging. To address this gap, 4 alternative science-based approaches have been established by EMA. The first one is the widely adopted method named the carcinogenic potency categorization approach, so the CPCA, which groups nitrosamines into potency categories with associated acceptable intake limits based on considerations related to structural alerts and to structural activity relationships. Another option is to use a negative result, for example, from a GLP compliant enhanced Ames test. And if the test is negative, the nitrosamine may be controlled according to that using the default TTC limit of 1.5 microgram per day. Of course, if the result is positive, an AI should be instead derived through the CPCA or other approaches. The read across is also another accepted strategy. So in this specific case, if a structurally similar surrogate, so an analog, a surrogate nitrosamine, has well-characterized carcinogenicity data, its TD50 value can be used to derive the AI of the target nitrosamine. And finally, if a nitrosamine gives a negative result in a well-documented in vivo mutagenicity study, it can be treated as a non-mutagenic impurity. And in that case, the impurity can be controlled under ICH Q3A or Q3B, regardless of any AI calculated using the CPCA, read across, or the enhanced Ames test. Next. We mentioned before that the regulatory authorities are well aligned on how to deal with nitrosamines. While the EMA and the FDA recommend broadly similar strategies for the assessment of nitrosamines, it's also important to keep in mind that there are few key differences that are worth noting. In relation to the CPCA classification, the approach is almost the same. In this case, the only difference is that the lowest limit is set to 18 nanogram per day by EMA, while it is at 26.5 nanogram per day by FDA. With respect to the enhanced Ames test, both agencies accept a negative result from a GLP compliant enhanced Ames test to support higher AI, but FDA typically requires additional data. With respect to read across, there is no significant difference among the two authorities, while with respect to in vivo mutagenicity studies, while EMA accepts a negative result from a well-conducted in vivo mutagenicity study to justify control as a non-mutagenic impurity, the FDA generally considered such data not sufficient alone to support an AI and the qualification thresholds defined in Q3A and Q3B. So it's generally used as in a weight of evidence approach as a supporting information. Next. As we mentioned before, when we are dealing with a nitrosamine that are lacking sufficient toxicological data, the first approach that can be employed is the carcinogenic potency categorization approach, which is a structure-driven framework that is used for nitrosamines based on their predicted carcinogenic potency when the compound-specific data are limited or lacking. This approach integrates and is the result of all the SAR considerations published in recent years by the scientific community and in the literature. And however, it's important to take into account that the CPCA approach is not universally applicable to all nitrosamine structures. This methodology specifically applies to the alkyl N-nitrosamines, that is, nitrosamines with an alpha carbon on both sides of the nitroso group, which share the same alpha hydroxylation activation mechanism. It does not apply to other nitroso compounds, such as, for example, the nitrosamines, nitrosoureas, the nitrosoguanidines, where the alpha carbon is double bonded to a heteroatom, nor to nitrosamines in which the nitroso group is attached to a nitrogen within a heteroaromatic system, the nitrosated indoles, for example. And the reason of that is that these specific compounds may follow a different mechanism of action. For those compounds that are containing more than one nitroso group, the acceptable intake should be based on the moiety with the highest predicted carcinogenic potency, so according to a precautionary approach. So, and finally, if more than 2 nitroso groups are present, regulatory authorities generally recommend to consult directly with them in order to identify and work together on the specific case. Next. As we mentioned, the CPCA approach assumes that the prediction of the carcinogenic potency of nitrosamine can be generated based on the assessment of its structure features, which are descriptive of the metabolic activation of the nitrosamines. And the key elements that are taken into account are the following 3 elements: the number of alpha hydrogens, which determines how readily the compound can undergo the alpha hydroxylation; activating features that can increase the likelihood of the metabolic activation. For example, specific electronic or steric environments may promote the formation of a reactive diazono ion. Deactivating features, which are structure elements that either they can hinder metabolic activation or enhance the clearance through alternative pathways. And each of these 3 components is assigned a score, and then the scores are summed to give a total potency score, which is then used to assign the nitrosamine to a carcinogenic potency category. Next. So in this slide, I show you the flowchart that is typically used to predict the potency category of a nitrosamine using the CPCA decision tree. So as you can see, the decision tree that is represented here combines a series of structured questions with the calculation of the potency score, which reflects the key structure-activity relationship features that we mentioned before. The flowchart can be applied manually, but is also implemented in software tools. So there are software tools where this workflow is automated in such a way that the process that can be facilitated and provide a consistent application. By following the decision tree and calculating the potency score for the nitrosamine of interest, the compound is finally assigned to one of the 5 CPCA categories, each associated with the corresponding acceptable intake limit. Next. Another approach that we mentioned can be, sorry, can you move next? I think you went back. No, sorry. Sorry. It's fine. I was just okay. That's fine. So another approach that we mentioned that can be applied when the nitrosamines are data poor is the read across. So this is one of the most commonly widely used methods to fill data gaps in human risk assessment, and this, for example, can be useful when there is the need to increase the limit with respect to the CPCA approach. So that is an approach that can be applied together with the use of an enhanced test or the use of in vivo genotoxicity data. Next. So the read across specifically involves estimating the potency of a nitrosamine by comparing it to a similar compound for which toxicological data are available. The justification of the approach is based on the evaluation of the similarities across different levels, as well as consideration on the underlying mechanism leading to toxicity. Next. This slide outlines the typical workflow for applying a read across. So the process typically begins with the identification of suitable analogues, often using databases such as, for example, the carcinogenic potency database. The quality of available carcinogenicity data for the analog is then evaluated, and the assessment of the similarity between the target and the analog is performed. This is really very crucial in the read-across approach, where this similarity assessment is performed considering multiple domains, multiple lines of evidence, starting from structure similarities, but also including physicochemical properties, ADME and the metabolic pathway similarity, and the reactivity and the biological and toxicological similarity. If the identified analog meets the criteria across all these domains and has a reliable TD50 value, then the data can be used to derive an acceptable intake limit for the target nitrosamine. So here is represented also an example where read-across was applied to the target compound, the nitrosamine, which was lacking sufficient robust carcinogenicity data. So in this case, a well-characterized nitrosamine, the NNK, was selected as an analog due to the shared features, the similarities in the metabolic activation and the DNA reactive profiles. And because NNK has well-established TD50 values, this data could be used to derive a perceptible intake for the nitrosamine. So to conclude, nitrosamines are a challenging class of impurities due to their potency and the potential for formation in a wide range of pharmaceutical contexts. And where substance-specific data are available, we saw that limits can be derived directly from the data that are available, while when substance-specific data are lacking, we can apply a number of science-based strategies. The CPCA is one of these, read-across, enhanced Ames tests, or in vivo data. And ultimately, the goal here is not really to eliminate all risk, but rather to define and manage acceptable risk. So grounded in toxicological science and supported by a transparent rationale that is aligned with regulatory expectations. So I thank you for your attention, and this is, I also would like to thank, of course, my colleagues for their support on this specific topic.
Thank you, Manuela and Caterina, for this interesting and valuable presentation. What is clear for me is that after this presentation is that managing a nitrosamine risk goes beyond the compliance. It's a multidisciplinary challenge requiring tight collaboration across toxicology, chemistry, quality, and regulatory functions. So thank you very much for your presentation. During also the registration phase, we received some questions that we have included in this last slide. The first one, I think, is for Caterina. So once a product has been manufactured and packaged, is it common that further nitrosamines get created over its shelf life? I think this was also clear along the presentation, but Caterina, if you want to add something, thank you.
Yes, so we have stability studies ongoing in our laboratory, and we have noticed that we could observe an increase of the nitrosamine during the stability studies. If, of course, at the time zero, there was a presence in traces of the nitrosamine, and during the stability with the checkpoint, usually until 36 months, we could notice an increment of the nitrosamine.
Thank you, Caterina. The next question was, I think, for Manuela. Is the less than lifetime approach applicable to nitrosamines?
Thank you. Well, traditionally, nitrosamines have been excluded from the application of the less than lifetime approach due to their classification within the court of concern and the assumption that even shorter-term exposures might pose a significant carcinogenic risk. However, this is a very key topic, and the recent scientific evidence challenges this specific view. In fact, there is a recent paper from Felter et al. It was published in July 2025, which examined the question on whether the empirical data that are available for nitrosamines are consistent with the principles underlying the less than lifetime framework, which is that the carcinogenic hypothesis is driven by cumulative dose, not by a daily dose or dose rate. And across a number of studies that were evaluated, it was found to be a function of the total cumulative dose rather than the daily dose. So these findings are consistent with the scientific basis of the less than lifetime approach that is outlined in the M7 guideline, which supports the use of higher acceptable intakes for exposure of a limited duration. So there are some uncertainties in this specific question, but regarding the potential for DNA repair mechanism, also we need to take into account that there might also be a risk of an overwhelmed if the lifetime equivalent dose is delivered over a very short time frame.
The next question was about how we can predict and how we will decide the limit of nitrosamines impurity in the product and API. I think that was also illustrated along the presentation of Caterina, but Caterina, if you want to add something more.
Yes, the limit of the nitrosamine impurity can be calculated with the acceptable intake, as I explained during the presentation, divided for the maximum daily dose of the product or the API. If the nitrosamine is not listed in the list of EMA or FDA, or the nanogram day, so the acceptable intake is not present in the list, I think that Manuela explained very well how to approach when we do not know the limit and how to calculate. From a validation point of view, from a technical point of view, we tend to validate the method at the lowest nanogram day acceptable intake, so 18 nanogram day, in order to start conservative. So once the nitrosamine is added in the list, or the limit is of nanogram day is calculated, we have already validated a limit as a worst case. So we are compliance with the lowest nanogram day present.
Thank you, Caterina. And this is for you, Manuela. Is CPCA applicable to all nitrosamines?
Thank you. Well, yes, as we mentioned, the CPCA applies to N-nitrosamines and not to, for example, nitrosamines, nitrosoureas, nitrosoguanidines, or nitrosamines that are contained in heteroaromatic ring system, like the nitrosated indoles. However, it's also important to keep in mind that the health authorities, such as, for example, the EMA, have started to provide AI values for a broader range of nitrosamines beyond the CPCA scope. So, for example, in Annex 1 of the EMA nitrosamine guideline, you can find a second worksheet listing acceptable intakes for other nitrose compounds, which includes the aromatic nitrosamines, nitrosoureas, and the nitrosoguanidines.
I think we have time also for one more question that was, yes, there were a lot of questions about the nitrates in the product. So, Caterina, this is for you, microcrystalline cellulose excipient. Do nitrates over shelf life of excipients? We perform this kind of test.
Yes, in my presentation, I did not report this analytical technique that we have in the lab. There is ion chromatography that we use for the analysis of nitrates. We do not have stability studies on nitrates ongoing, but we perform analysis on excipients regularly to check the presence of nitrates.
Sorry, I was in mute. There were some other questions also for Manuela. Are the limits set by health authorities based on CPCA?
They do use a CPCA, but health authorities also publish limits that are not strictly based on CPCA. So, for example, EMA published limits of 1,500 nanogram per day based on negative Ames test. This, for example, is the case of the nitrosambroxol. There are also limits that are based on read across. There are compounds where the limits are published by EMA stating that the nitrosamines is non-mutagenic impurity based on in vivo negative studies. And there are also non-mutagenic impurity based on a read across in EMA.
Okay, thank you, Manuela. There was another one that maybe is linked to the previous. Can I apply read across to increase this limit?
Yes, as we mentioned before, read across is a valuable tool, is a valuable approach for deriving safety limits to increase the limits when the experimental data are available or when the CPCA limit is too low. It relies on a very robust integration of structural activity relationships, mechanistic insights, experimental evidence to predict the toxicological outcome. My personal view, if I can just add a comment here, is that in the context of nitrosamines, regulators are prone to express skepticism due to the complexity, the variability, and the potent carcinogenic potential of this chemical class. So it's something that is there, is defined and suggested by the guideline to be applied carefully and taking that position from regulatory authorities into account.
Thank you very much. There are some other specific questions that might require to gather some information. So it was about anhydrous system oil. Do nitrosamines feasible to form, or nitrosamines form only in aqueous formulation? As far as we have seen so far, I thought as a chemist that as long as we are dealing with nitrosamine, I thought that, for example, solid-solid reaction was very unlikely to form, but with this nitrosamine along these studies that we gather so far, is that is possible. I don't know, frankly speaking, I don't have an exact answer to this question, but I can gather some information in order to be more precise on this specific question. Yes, there was another one for Caterina. I don't know if you can answer to this. What kind of excipients that you are mentioning in your case study, excipient 1, excipient 2, were used in your compatibility studies with the API?
Once was micro crystalline cellulose, and the other one was another excipients were with a very low content in nitrates.
Caterina, do we have method for testing nitrates in cellulose product with LOQ below 0.05 ppm?
LOQ below 0.05 ppm is very challenging for IC chromatography. We do have IC with mass spectrometry, but not in pharma. But we have the possibility to test it. Usually we are around on the sample 1 ppm, 0.5 ppm. But because the dilution factor has to be high, so we cannot go very low with standard concentration. So but we are testing. We are testing to a preparation that can go lower in sensitivity.
Also, cellulose is a very hard matrix.
Yes, you can't work concentrated. You have to work a little bit diluted. So the standard, we are around 5 to 10 micrograms liter on the standard concentration. So working with a dilution factor that has not to be so small is difficult to reach 0.05 ppm.
Okay, thank you. I think we have time for the last question that was for me, that I think I can answer to this one. Is the compatibility study must be performed on finished product, dosage, form, or powder blend? The case study that Caterina presented in their speech was a specific problem for a product. We noticed formation of nitrosamines in the finished drug product along with the time and the shelf life. So in collaboration with these customers, we performed a compatibility study on the specific excipient with the API in the same quantity and same ratio they occurred in the finished drug product, in order to see which excipient could give a major could lead to the formation of nitrosamine and in which magnitude. So it's a protocol that in some specific cases, as described by Caterina, was performed in our laboratory on input of our customer that had this kind of specific problem. Yes, it's not on the tablet. We made it on the blended, just to complete the question. So there were dual mixing on API and single excipient to check what was at major risk of lead to the formation of nitrosamines. Okay, I think we have finished our time. If there are more questions, we can address the answer. If yes, we can answer to them in the next day. These are our main contacts, so don't hesitate to contact us for any further request of information and support. So thank you, Caterina, thank you, Manuela, and thank you for all the attendance today.
Thank you.
Bye-bye. Thank you.
Bye.
Thank you. Bye.